That's what it said.
Hello, my name is Madison Lowrey, and I decided to research the effects of alarm pheromones on Apis mellifera appetitive learning. I will begin with an introduction of alarm pheromones and defensive behavior in Apis mellifera, and then I will explain what appetitive learning is and how we test bees' learning abilities. Finally, to end this presentation, I will describe the results of the experiment and how appetitive learning in Apis mellifera was affected. I will begin by introducing the western honeybee, Apis mellifera, which was the type of bee used in the experiments testing appetitive learning with alarm pheromones. Apis mellifera is the most common bee used in beekeeping and is essential in our agricultural economy. Apis mellifera is a eusocial species that relies on chemical signals, such as pheromones, to communicate among one another. In order to understand the importance of this experiment, it is crucial to understand how Apis mellifera uses alarm pheromones in defense. By understanding the findings in this experiment, the behaviors of bees can be more deeply understood and will help us create a better environment for bee populations to thrive. An alarm pheromone is secreted by Apis mellifera when they encounter a threat that could harm them or the colony. This pheromone recruits other bees from the colony to help defend against whatever the alarm pheromone has been placed on. There are many different types of alarm pheromones with many different components. Studying the different components of certain alarm pheromones will help us understand why they react the way they do when exposed, as well as how they react. The specific alarm pheromone used in this experiment was the sting alarm pheromone. The sting alarm pheromone is a complex blend of 40 components that triggers an aggressive fight or flight response in bees who are exposed. One component of the sting alarm pheromone is isopentyl acetate. This is the main component of the sting alarm pheromone, and the amount released by bees increases with age. Another interesting characteristic of alarm pheromones that is critical to understanding Apis mellifera behavior is that when the alarm pheromones are released into a target, this increases the chance of the target being stung again. In the experiment performed, isopentyl acetate was the compound used to test the target being stung. Test the effects of Apis mellifera learning, since this one component elicits the same response as the entire blend of components does. It was found that exposure to isopentyl acetate impairs appetitive learning in a dose-dependent manner. This impairment of appetitive learning turned out to be long-lasting when exposed. IPA was able to inhibit learning for up to 24 hours after exposure. Next, I will transition to the defensive behavior of Apis mellifera. By breaking down the events of a defensive response, we are able to determine the facts that regulate each behavior. As you can see in Figure 1, there are many pathways in a defensive response. They all eventually lead to attacking or disengaging. Another hormone, called the juvenile hormone, also plays an important role in priming workers for a defensive response. Appetitive learning is a form of associative learning used by Apis mellifera and many other species to relate resources with specific aromas. Without this learning ability in honeybees, they would be unable to associate floral aromas with the presence of pollen or nectar. An example of appetitive learning is the proboscis extension response conditioning. In this conditioning, a honeybee is held and a pipette filled with sucrose is set in front of the bee just outside of reach. The bee will extend its proboscis to the sucrose and therefore learns to associate a stimulus with a sugar reward. This technique of conditioning was used in the experiment to test Apis mellifera abilities to learn even after exposure to an alarm pheromone. Now that I have discussed all about all the information important to understanding the results of the experiment, I will discuss the conclusions drawn and the data that supports these claims. In the experiment, it was found that IPA was the main component of the sting alarm pheromone that decreases appetitive olfactory learning. This was tested against an untreated group and a group treated with oil. As seen in Figure 2, the red line represents the group of bees exposed to IPA and their percent response to the conditioning after. It is clearly much lower than the other two groups, suggesting IPA indeed does decrease Apis mellifera's ability to be conditioned. Overall, the data shown in figures 2 and 3 both indicate that appetitive learning in Apis mellifera is indeed affected by IPA exposure. The longer the bees were exposed, the longer the effects of the IPA. These results may have arose because when exposed to an alarm pheromone, Apis mellifera will not stop until given the signal to disengage or is killed. Foraging and other tasks become secondary priority when behind the object targeted with the alarm pheromone. Another variable to consider when drawing conclusions about effects on appetitive learning is age. The age of honeybees tested was held constant since older bees are much better at learning through conditioned responses than younger bees. This concludes my presentation on the effects of alarm pheromones on Apis mellifera appetitive learning. Thanks for listening.